Good morning or good afternoon. Well, wherever. Let's do it. Our last day. Well, thank you all, Cesar, Woodson, Mariana, Jones, to make this dream possible. It will be nice to listen to you. I will be behind the cameras again. So our last day, keep your brain boiling, the brainstorm happening, and let's do it. Thanks a lot. Let's listen to these wonderful presentations.
Welcome to everybody for our last day of spring school. As the other day, I hope that we have a great discussion, and we have first all questions we welcome, okay, in Portuguese, in English, and please write all of them and send to us using the YouTube chat. And today we have lectures to meet that I'm so difficult, so I will have many questions to do in the end. Today we have lectures about biogeography and macroecology of fishes in South Atlantic with Dr. Hudson Pinheiro. Dr. Hudson obtained a PhD in ecology and evolutionary biology at University of California in the United States. We will have lecture 2 about functional diversity in marine forests of South Atlantic with Dr. Marina Bender. She will start with us. Dr. Marina Bender obtained a PhD in ecology and conservation at Federal University of Paraná in cooperation with University of Montpellier in France. Oh, good. And to the end, we will have a lecture about the diversity patterns of echinoidea in marine forests with Dr. Cesar Cordeiro. Dr. Cesar obtained a PhD in marine ecology at Federal University of Fluminense. It's nice to stay with you today. You can say hello to everybody to next to us. Say good morning, good afternoon for everybody. Or no, we can start again with Mariana, I'm sorry.
Good morning, everyone. Thank you for the invitation. It's a pleasure to be here and to be here with Hudson and Cesar, which are colleagues that we've been colleagues for a while. Thank you. It's an honor.
Okay, Cesar, you want to say something? Say hello to everybody. Say good morning.
Hello, good morning, good afternoon. Very happy to be here. Very happy with the invitation. So I'll be the last one, and we'll listen to the brilliant talks of Mariana and Hudson first.
And you, Hudson, say hello for everybody that can listen to us now.
Hi there. Hello, everybody. Thank you for being here. And yeah, today we have some great presentations. It's an honor to be here with Mariana and Cesar. Hope you enjoy the presentations.
Oh, so good. So let's go. Mariana, we will start the lecture, and the time is yours, Mariana. And on the end, next to the end, I appear here now, so 5 minutes. And I will need to that Cesar and Hudson turn off the camera to internet connection, and I will turn off my too. So Mariana, I'm here on the backstage, so anything you can say me that I'm back. So good lecture.
Awesome. Thank you. I'll try to share my screen with you. So let me know if everything is working perfectly, please. Because we did not test this before, so let's see if it works.
Yes, I will stay with you before we start.
Okay. I guess we're starting.
I can see your screen.
Okay, let's hide this. Sorry, guys. Okay.
Okay, good lecture.
We just missed, Mariana, the full mode, you know?
Yeah,
we
yes.
are observing the edit mode, just.
Let's see. Can you see it now?
Yes, it's perfect.
Oh, okay.
Thanks a lot.
Okay, guys. Good morning or good afternoon for everyone who's out there. I'm glad to be here. I would like to thank you for the invitation. It's a pleasure. If you have any problems with connections, if my internet is not working properly, please let me know. So you just send me a message on WhatsApp. I know some of you guys have my contact. Okay, so let's do this. My name is Mariana Bender, and I'll be talking here today about functional diversity in marine forests of the Southwestern Atlantic. So I work mainly with functional diversity related especially to reef fishes, but I extended this talk a bit to explore functional diversity for other marine organisms, mainly vertebrates. And also the focus here is the Southwestern Atlantic, but you also find some examples for other parts of the Atlantic. So the entire Atlantic will be here in my presentation. So I'd like to thank you for the invitation. It's a pleasure to be here. And what you see here in my talk, we'll start with functional diversity. I'll explain a little bit about this for you guys, and we'll talk about later on if you have questions. Then we'll take a look at what we have been considering as functional traits. We'll take a look at traits for fish and benthic organisms, the form, the benthos. And then we'll take a look at patterns for functional diversity Southwest. Many, but I focused in so the focus is mainly the Southern Atlantic. So diversity is from other diversity facets as taxonomic diversity and phylogenetic diversity. So functional diversity is a part of the diversity we can find in ecosystems, communities, or populations. It exists in different levels of the organization of the ecosystems. So for functional diversity, what we can consider as as characteristics of, we use, let's take a step back. Usually we take a look at communities from a taxonomic perspective. We consider if a community is rich in species, its composition, but we can also consider how species vary in their functional characteristics. Okay, what are these functional characteristics? What we call traits. These characteristics are the way in which species interact with each other and the way in which they interact with the environment. So this is why traits are so important, because if a fish is consuming algae, for instance, it is interacting with this algae species through its functional characteristics, through its traits. It's biting on algae using its teeth, its mouth. It's using its tail to swim in different parts of the water column, and the algae is using its traits herself to grow in a different way or in a different direction. The algae growth will determine its position, its direction, its form. So we consider different characteristics of the organisms because they are so important to shape the way they interact with each other. And you also see that the way they interact with each other will influence how these traits exist in the tree of life. You can think of the tree of life for algae, for coral species, fish species. So what we consider usually when we talk about traits, we consider the form of the species, their performance, if they, for instance, for fish, if they bite too much or if they don't bite too much on algae or other organisms. Diet composition is another trait that we use, growth and behavior. So these are all aspects of functional diversity. And you can also think of these different aspects of functional diversity or traits for fish organisms, okay? So they have their own characteristics. What we usually consider for fish, for instance, This is a work from Kim Bio and collaborators, which was published this year. So for fish, there are a number of characteristics considered as range, mobility of the organism, activity is mainly at night or at day, the position in the water column, if it's associated to the bottom or to the high levels in the water column, if it has a tail that it's of one form of the other. So so these are the different traits that we use, and we usually combine these different trait categories to associate species or distribute species in different functional groups. So if a species has large body size and is a piscivore and swims in its activity is mainly diurnal, then this species will belong to one functional group. While herbivores with larger body size that swim that are associated to the bottom, for instance, will form a different functional group. But why do we study traits? Why are they important? Because mainly, as I said before, traits are important links. They work not only at the ecological scale, where we consider the current scale that we're working, because they influence if species will interact with each other within one manner or the other. But they're also very important because these interactions between species, which occurs throughout their traits, will determine if traits will be maintained in the tree of life, or if they will disappear in the tree of life. Okay, so they are important links between what happens in the ecological time scale and what happens in evolutionary time scales. We can talk more about this later on because we have limited time today. So basically what we use as I said before, what we use to categorize species into functional groups, for instance, are their traits. Okay, and a common metric that is used to assess or measure functional diversity is the functional space. So how do we measure this? Well, we considering the traits of species. For instance, here I use parrotfish. You can imagine these parrotfishes have different body sizes. Okay, they have different diets, and they have different mouth positions and different tail forms. So considering the set of traits, we measure the distance of species considering traits, and we distribute species in a functional space. And we then measure the dimensions of the space as a proxy of the functional diversity of this community. And there are also other metrics that consider how species occupy this functional space. So basically, we're working with the distance and difference between species in this functional space. So this is an example for fish, but this is also used for, benthos, and we can measure how organisms that form the benthos that contribute to the diversity of marine forests, we can take a look at how they occupy trait space. And here we'll have different traits for fish and different traits for benthos. For benthos, you can think of growth form, size, and traits associated to reproduction, for instance, and then we'll take a look and understand how they are distributed in functional space. Okay, but then you can ask me, what is going on in the Southwestern Atlantic? What is like functional diversity for marine forests, or mostly reef ecosystems in the Southwestern Atlantic? So here we'll be concentrated at this region of the Atlantic. Okay. Sorry guys, there's lots of effects and the Southwestern Atlantic remains there in the background. Well, first I'll start with an example that it was developed on top of this framework here of Slater and collaborators from 2008. This work of Sergio sets the base for what is the biogeography of reef fish in the Atlantic Ocean. So we had this understanding that there are different biogeographical provinces within the Atlantic. This is for reef fish. So we have the Western Atlantic very well separated from the Eastern Atlantic, and you can see here that the Northwestern Atlantic is also separated from the Southwestern Atlantic. But these biogeographical regions were delimited considering reef fish species occurrence, presence and absence data. I believe that Hudson will talk more about this, but considering that we have this baseline here of the biogeographical regions, we wanted to explore for the Atlantic what are the relationships considering the proportion of species within different families across the Atlantic, and the proportion of species that have different traits as body size and trophic categories. So considering body size and trophic categories, what are the relationships between different communities across the Southwestern Atlantic? So we did this in 2013, a while already. This was part of my PhD. So we look at the functional structure across the Southwestern Atlantic, exploring body size and trophic categories. I'll focus here on body size, okay? So what you see in red over there are communities that are characterized by biogenic reefs. So we have biogenic reefs and reefs of other formation as rocky reefs, for instance. So what we expected here was that reefs that have biogenic formation would have different trophic structure and different body size structure relative to other reefs that have other formation. And that these reefs that have biogenic formation will work as a filter to different types of species with different characteristics. So we used here a cluster analysis on the proportion of species with different body sizes, trophic categories, and within different families. I can talk more about the analysis later on our discussion. And what you can see here is that we rescued 4 families, different groups of sites. Okay, so we have sites that were grouped here based on the proportion of species within certain families. And here are highlighted in different colors the different parts of our cluster. Okay, so for instance, in this region of the Caribbean, you have a greater contribution of serranid species and gobiids and labrid, for instance, labrizomidi, for instance. While as we move towards other regions of the Atlantic, you have a greater contribution of other families. For instance, Carangidae here has a greater contribution in regions as St. Paul's Rocks, Ascension, and Santa Helena. Okay, so you see that this varies across the different communities. And if we compare this cluster, the cluster structure with what we have from the biogeography and evolution, which is the paper from Sergio, you Atlantic islands in the mid-Atlantic region coded in green. So we have this separation. So for families, you have the imprint of biogeography. Okay,
Mariana.
Okay.
Just sometimes your audio is cutting a bit, so if you can, please just a little bit slowly.
Okay,
just
sorry.
to avoid. And we have a simultaneous translation, so just to facilitate on the other room. Thank you.
Okay. If you want, I can move to my living room because I'm in my office. So if the internet is poor, you let me know.
Up to you. I think is okay. I think we can continue. Just try to speak a little bit more slowly.
Okay. So going on with this example here, we have the results for body size. So basically, we're looking here at the contribution of species with different body sizes for different communities of reefs across the southwestern Atlantic. So here we did not rescue the imprint of biogeography as we did in the family example. What we have here is that the great contribution of species with small body sizes, here are small body sizes, okay, in this region of the Caribbean. And as we move towards isolated areas that are very distant from the Caribbean, for instance, in the mid-Atlantic ridge, and in the Eastern Atlantic, for instance, in São Tomé, we will rescue here the greater contribution of large body fish. So this means that in these communities, there is a greater proportion of species that have medium to large body sizes. And this is mainly the influence, and this rescue mainly the influence of isolation and the distance from the center of diversity in the Atlantic, which is the Caribbean. But when we are in communities that are very close to the Caribbean, that are within the center of diversity, we have a greater proportion of small species. Okay, relate to the space at biogenic we have complexity.
Mariana, now we lost almost everything.
Oh.
Guys, is this just for me or everybody are lost in Mariana too?
For me, I'm lost in Mariana too. Camera set.
Let's see if we can connect with Mariana again. Maybe if Mariana move to another room. Hudson, can you check if Mariana is okay? She fall. Yeah, that's an interesting aspect. Well, I'm sorry, I forgot an important thing. Mariana is back.
Hey.
You are back. However, let me check your audio, etc. Nope. I can't see you properly here. I don't know why you don't appear to me. Can you listen me, Mariana? No. Well, I think Mariana will try again to connect because she's well, the oscillation of the sign, I don't know what is happening. But I forgot to say, well, in our program was expected the presence of Dr. Marina Cicini. However, Marina got sick last week and she couldn't come. So I will, at the end of the presentation, I will try to bring some of the aspects of biogeography and macroecology that was expected in the Marina's presentation, just to fill with this content our discussions during the regular course. So let's try again, Mariana. Let's see. Let me see if I can see you here among us. Oh, seems we have a connection problem, a huge connection problem. Yeah. Well, okay. Mariana is trying again. I think we can try another time. If Mariana can't stabilize the connection, I will suggest to move to the next presentation. Then we have more time to solve Mariana's connection. What do you think? Cesar, Hudson?
I'm available here. If Mariana can connect, like, we can go ahead.
Yeah, seems she really have a big problem. Just because she disappeared off the room. Yeah. Let me check our connection with the YouTube. Everything's fine. Please, guys, say us in the chat in the YouTube how is the connection for you, the image, the sound, everything. That just to let us up to date with that. Marianne is back. Let me see. Yeah.
Hey guys, sorry.
No worries. This is a pandemic.
Yes, we have another
pandemic troubles. Yeah.
I was so excited talking, and then I had an issue here with internet this morning. The light, we just lost electricity here, so probably that's the issue.
Yeah.
Okay, I'll try to get back. I don't know how many minutes I still have.
Well, man, take your time. Let's see. Try to finish as possible as you can. But
okay. So I have poor light here in my living room, so you have to. My face is not very. You cannot really see me properly. Okay, I'll share my screen and we'll do this.
Yeah, let's try.
I feel bad.
No worries, man. Man, this is a kind of normal thing. You see, man, every week. Almost every day I have this kind of problem. I think we lost Mariana again. No?
Yeah.
No, I'm here.
Are you there, Mariana? Oh, okay, good.
I'm just trying to share my screen, but I don't know. Am I sharing
sorry. I need
like...
to provide you the presentation mode, and now you can share your screen.
Okay, cool.
Mariana, this is happening every week. Well, you are teaching also, so we need to deal with this kind of thing all the time now. So no worries. I will turn off my camera just to facilitate the transmission.
Okay.
So good luck. Good luck.
Okay, if I lose connection again, then Cesar or Hudson can go on and present their talks.
Now every little thing's gonna be alright.
Thank you for your nice
good vibes, buddy. Good vibes.
Thank you, guys. Okay, so I was talking about body size proportions, communities of reef fish in the Atlantic Ocean, and you see here is the entire Atlantic. It's only the southwestern. But what I was saying before is that in the eastern, the Caribbean region, we have a greater contribution of small body species. And as we move to areas, which are center of diversity, we will find a greater contribution of large body species, medium to large body species. So these graphs here, they show the proportion of species within different body size categories. So what do we have? What's about these coral reefs that we have a greater proportion of small-bodied species? Well, in these reefs you have greater complexity. These reefs have this three-dimensionality, larger complexity, greater coral diversity, and greater diversity of benthic organisms. So you have greater opportunities for small-body species to survive, not only to survive, but also to the speciation of small body species, okay? And in isolated reefs, you have a greater contribution of large body species because large bodies are also a trait that helps species to cross geographical barriers. So large-bodied fish will cross and cross these barriers and have a greater colonization capacity, and also greater chances to survive in these isolated areas, okay? And you can see also not only the cluster analysis show this, but when we develop a redundancy analysis that relates the proportion of species within different body size categories with environmental characteristics of these communities, we'll see that small body species are indeed associated with larger sea surface temperatures, larger coral area, and coral richness, while large body species are mostly associated to these isolated areas and the distance from the center of diversity. So basically, if I'm a large body fish, I'll have greater chances to live in these isolated areas, to find partners to reproduce in these isolated areas, and also to cross barriers. So we also have other results for functional diversity. This is also related to the entire Atlantic, but I'll show what has specifically in the Southwestern Atlantic. And this is for betta diversity and functional betta diversity. So we have this work which is under review, where we explore the patterns of taxonomic betta diversity, phylogenetic betta diversity, and also trait or functional diversity, if you prefer. What happens here for betta diversity is, when we look at taxonomic and phylogenetic, the most important component of differences between reef fish, this is for reef fish, I forgot to mention, reef fish communities. The most important betta component for taxonomic diversity and phylogenetic diversity is the turnover component. But when we look at functional betta, the most important component is the nestedness component. This means that for the trait composition or functional composition, these communities are highly nested within each other, and they also have great similarities in composition. And if you take a look at these arrows here with different colors, they point to these communities that have similarities in betta diversity for their nestedness component. So the Caribbean has very similar values, the Southwestern Atlantic has very similar values, and the Mid-Atlantic ridge here has similar values and similar composition. So for the functional structure, it is very similar for reef fish across the entire Southwestern Atlantic, with the exception of islands. Okay, so this is the I wanted to highlight here from these maps. And this is for the turnover component. And when you look at the turnover, you can see where lies the differences between communities, which are in certain regions here. I'll move on because I have more stuff to show you. So for there's another work here for the functional diversity, and is also for the entire Atlantic. And this is for vertebrates that occupy reefs in the entire Atlantic. So vertebrates that can be found in reefs. And we're talking here about mammals, sharks and rays, bony fish, and turtles. So this is a work that is also under review, and we basically explored what are the ecosystem functions of these different groups, different taxonomic groups, and if these different species of very distantly related taxonomic groups, if they can have similar functions in these reefs. If, in other words, if they can perform redundant functions in reefs. So this work was, for this work, we compiled information for 224 species of different groups. And in this graph here, what I want to highlight in this bar plot is that different groups will contribute to different functions. And you have bioturbation, for instance, in this traffic regulation in this other side, okay? And in red here, you can see the proportion of species within these different groups. So we have the number of species that are threatened, and here on this part of the plot, on the bottom right, you can see the proportion of species within these trophic groups. And here I wanted to take a look at this bar over here, which is the proportion of top predators that are threatened in Atlantic Ocean reefs. Okay. So in this work, we mapped the species that occur in reefs that have these different functions. And you can see this is the pattern of species richness. So still the Caribbean has a greater proportion of species richness, and the Southwest has a lower species richness, but still richer communities relative to the Eastern Atlantic. And when we map functional richness, we rescue the same pattern. So communities in the Caribbean have greater functional richness relative to the Southwestern Atlantic, and the Western Atlantic is richer in functions relative to the Eastern Atlantic, okay? We also map other functional indices, and here you see functional uniqueness that is basically we can consider as the opposite of redundancy. And we have across the Atlantic very similar unique, it seems that we have similar uniqueness values, which means we have communities that have unique functional contribution of different species. And what we wanted to do in the next step of this work was to take a look at species that belong to different trophic groups. And if taxonomic. We have species that are mesopredators, are fish, or if there are sharks or rays, or if there are mammals, for instance, can they perform similar functions? What happens if we remove species from these groups? What happens to the functional structure of the Atlantic? So this is what we have done in this analysis, and we have selected 4 different functions: bioturbation, herbivory pressure, which is on B, the function of marine top predators, and the function of mesopredators. And we removed species that perform these functions. We first removed these ones. We first removed herbivore, marine mesopredators, and then removed other species randomly. And we contrasted this with a new scenario. And what we have found that I want to highlight here is that when we remove marine mesopredators, we have a greater loss in functional richness than expected by chance. This means that these marine mesopredators contribute to a large part of the functional space, or with a large proportion of functional diversity to these reefs. And here you can see that the line is below what is expected at random. So if we lose these mesopredators, we'll lose a greater part of functional diversity in these Atlantic Ocean reefs. Okay. And when we map this, sorry, I'm going too fast, I still have some other results to show you. And when we remove these species from communities, all of these grid cells are considered the assemblage or the community level, okay? This is the proportion of species removed, and you see that in green, there are 20 to 30% of species removed, but the functional loss reaches 90%. In red, this is the functional loss of communities when mesopredators are removed. So this means that if you lose 30% of species, you can lose up to 90% of your functional diversity in these reefs only from removing mesopredators. So this means that these reefs are threatened because this is only species richness. Because we are fishing top predators, we're removing top predators, and now we're going to the mesopredators. We're already eating these species, okay? And now I'll move to another example of Anaïdi Awet, this paper where she explores the diversity of benthos organisms. This is the community composition this is the composition for benthic communities in the entire Brazilian coast and oceanic islands. And here you see in these different bars, the contribution of different organisms to the total cover of the Benthic community. And you see in green that these reefs are largely dominated by algae, turf algae, and macroalgae. And I am certain that Paulo will talk more about this later. But I wanted to make a link here between what we can see for the composition in terms of species or the taxonomic composition. And here we have a separation from reefs that are dominated by macroalgae and reefs that are dominated by turf, here in this part of the graph. But what NIEG is exploring that is in preparation is the relationship between these communities in terms of functional diversity. So this is basically a map of the functional diversity of benthic organisms in the Brazilian province. And you can see that there are certain parts that are greater contribution of carnivores and other bioconstructors up here. So she mapped the functional diversity for Brazilian reefs, and what she has seen is that across communities, functional diversity here is not similar. There are certain places concentrate a greater contribution of functional diversity, and here specifically is Espírito Santo. Guarapari is mapped here in this black rectangle that I highlighted, and other communities have subsets of functional diversity. This means that the benthic communities are not similar across space here in our coast and oceanic islands. Okay, and she explored in addition to functional richness, other functional indices, which I'm not going to comment here, but I'm now moving on to the final part of my presentation. And here I want to show some of the results that are in line with those of NIEG AWET. And here is a study that is part of the reef synthesis working group. And sorry, this work is led by André Luza, which is the postdoc of our synthesis group. I'm very sorry. I have a loud dog. And this work was done for fish and benthos organisms in conjunction. Melissa. Okay.
Beautiful name, indeed. Indeed, beautiful name, Melissa.
I'm sorry, guys. Okay. So
Good girl.
So fish and benthos together. What we have done here in this work was, because until today we haven't explored if these different parts of marine forests, if they vary in the same direction, if they have similar functional diversity in space, in same sites that you have large functional diversity of reefs, if you have large functional diversity of coral and other benthic organisms. So what we did here was to test the congruence in functional diversity, different metrics, but here I'm going to focus on functional richness, and the response of these different functional metrics to environmental drivers. And the environmental drivers we have used are temperature, turbidity, depth, and isolation measured as distance from the coast. And what we have found is that there is large spatial variation in functional diversity for both fish and benthos, and that these different groups do not respond similarly to environmental drivers. This means that benthos and fish will not respond similarly to depth or isolation or turbidity. They will respond differently. And here what I want to highlight is, sorry, I forgot to highlight with the rectangles, but Espírito Santo hosts the largest contribution of benthos to the reef diversity, as NIEG has found. And for reef fish, sites in the southern limits, as Alcatrazes, for instance, host a large proportion of functional diversity of fish communities. And we also found that they are not, these indices are not similar across space. You can see here in this matrix of correlations that you have mostly negative correlations, and the ones that are significant are in bold font here. So some of them are in bold font and are significant. For instance, functional evenness of benthos and functional richness of fish. So we found no congruence. And I'm sorry, but I have to end my presentation with my loud dog in the background. And these are mostly the results that I wanted to share with you related to functional diversity. And I would like to thank the invitation of Marina and Paulo to join this spring school. It was great. I would like to express my gratitude to all my collaborators, people that are here sharing the talk with me, and other collaborators around the world and also throughout Brazil. I'm very happy to be part of your research groups. And that's it, and I'm open to questions when we end. Thank you guys, I'm sorry for all the problems.
No worries, no worries. So it was really very interesting. Well, very, 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 very colorful. I love the fishes, all the connections.
I have questions.
That's great, that's great. Let's move, Jonas.
To the end, yes? Our questions going to be to the end? Yes?
Yes,
okay.
Yes, let's do it on the end.
Mariana.
Yeah.
Now the Cesar, yes? Or Hudson? Who come now? It's Hudson, yes? Hudson?
Next is Hudson, yeah, I think so.
Where are you? Turn on your camera, please. Oh, yeah.
Hi there.
So, Hudson, the time is yours. I'm here. Anything and problem that you have? Wait a minute. You can see on your screen sign up more, and that you can share your screen. You can do it just now. Oh, your current time is yours. I back here next to the end. Okay. So good for lecture to you.
Is everything okay? I can't see myself.
Yes,
or
I can
see.
Yes, it's so perfect.
Okay, so thank you again, for the invitation. My name is Hudson Pinheiro. I'm a researcher of the Center for Marine Biology of the University of São Paulo, and I'm also associated with the California Academy of Science. Today, I will talk about the biogeography and macroecology of fish in the southwestern Atlantic. So, I will start my presentation talking a little bit about evolution in the Atlantic and with him the southwestern Atlantic. And then I will talk about biogeography, a little bit about phylogeography, and finish with some macroecology patterns in the southwestern Atlantic. So a little bit as introduction, we have a very well-known pattern of biodiversity gradient that has a peak in this region that is known as Coral Triangle. Is a region that is situated between the Indian and the Pacific Oceans. And this has a very interesting gradient that runs both latitudinally and longitudinally as it gets distant from this center of biodiversity. And it occurs not only for reef fish, but also for corals and many other marine organisms. And another point that is very interesting is that we have a secondary center of diversity in the ocean, in the Atlantic Ocean, that is situated in the Caribbean, and it decreases towards the Brazilian coast and then to the East Atlantic. So I would like to highlight three very important points about the evolution of the biodiversity, the reef fish biodiversity in the Atlantic. One is that the biodiversity in the Atlantic evolved in a very highly isolated from the other marine provinces. It's very different from the Indo-Pacific, for example, where many lineages were exchanged between the Indian Ocean and the Pacific Ocean. The second point is that in the Atlantic, we have very high origination rates that were as high as those found in the Indo-Pacific. However, when we look at the extinction rates, we had a much higher extinction rates in the Atlantic. So from all that species and lineage that were originated, very few of them oceanic. So in comparison with the Indo-Pacific, we have today a very lower amount of species in total. So, and it's not only in the number of species, but also in functional traits too. This is analysis looking into functional traits of herbivores, and inside the dashed lines were the functional traits that existed in the Eocene, functional traits that were found in the fossil record in the Tethys. Tethys Ocean, the Tethys Sea, was a passage, was a region that existed between the Europe and the Africa and connected the Indo-Pacific and Atlantic in the past. So researchers found there many forms of herbivores, and we can see here that these forms in the left in blue, they expanded, they diversified in the Pacific, while they went largely extinct in the Atlantic. And today we found in the Atlantic only a subset of forms and functions in herbivory from what is found in the entire world. So moreover here, I would like to highlight three major patterns of connectivity and evolution that happened in the Atlantic. The first is the connectivity that existed between the Atlantic and the Indian Ocean in the past, in the Eocene, that comes all the way to the Pliocene, where we had this connection mainly through the Tethys Sea. So we have like today strong phylogenetic signals in many species that are found in the Atlantic, where the common ancestor is found in the Indo-Pacific, but this common lineage dates like over 12 million years old. That was the date when the Tethys Sea closed. So the second pattern is more recent. After the closure of the Tethys Sea, we have some lineage that invaded the Atlantic through the passage between the tropical Eastern Pacific and through the South Africa. So we have many lineages in the Atlantic that are that share like common ancestors with the Indo-Pacific, with the Indian Ocean and the Eastern Pacific, but they are more recent than 10 million years old. And we have more recent patterns with the Atlantic highly isolated, with exchange between biogeographic provinces inside the Atlantic, and very, very few lineage that enter in the Atlantic through the South Africa. So all these evolutionary process, they shape the composition of fish that we find today in many biogeographical provinces. Here we can see that the Southwestern Atlantic, as Mariana showed before, it's closely related with the composition that we found in the Caribbean, in the Mid-Atlantic Ridge, and is also very closely related to the Eastern Atlantic. And a point that I would like to highlight here is the position of the tropical Eastern Pacific. Although it belongs to the Pacific, it is closely related to the Atlantic biodiversity. It's because of the passage that existed before the closure of the Isthmus of Panama that happened 3 million years ago. So we have this historical biogeography influencing the composition of fish that we found today in this region. So inside the Atlantic, we have the process that regulate the compositions, the distribution of the biodiversity, and the evolutionary process are mainly related with major biogeographic barriers. We have here the Amazon and Orinoco rivers plume that separates the Great Caribbean from the Brazilian province. We have the Mid-Atlantic barrier, that is the distance of open water that separates the Brazilian, the Western Atlantic to the East Atlantic. And we have the Benguela barrier, that is a cold water upwelling that happens here that constrains the entrance of migrants from the Indian Ocean into the Atlantic. So one recent analysis showed a very interesting pattern of where different ecological traits, biological traits of the species, of the fish species, influentiate the possibility species cross like these barriers. Mariana told a little bit about that. So some are related to the rafting behavior, to the larval developing mode, body size, latitudinal range, and the ability to use multi-habitats. These characteristics allow the species to cross biogeographic barriers and to be present in different provinces. So starting with the Amazon barrier that separates the southwestern Atlantic province separated to the Caribbean province. It's very interesting because this is considered a soft barrier during the current sea level today. We have the fresh water that covers most of the shallow habitats, but we still have like deeper habitats, the mesophotic reefs that that are found below this plume. Here, these pictures below are photos taken of this reef that is situated below the plume. So there are many reef fish that occur in these deeper depths that are able to cross and to inhabit this habitat there. However, during the Pleistocene, during the ice ages that occur in most parts of the Pleistocene, the sea level was much lower than the current one. So this habitat was reduced a lot and were only found in the walls habitats that are, so it's like some data show that the shallow habitat was reduced in over 90%. So this constrained the passage of the species between the two provinces. So many species, including species that occur at the depths of fish, they were able to cross at certain moments, but in other moments, this passage was closer for them. So here, for example, we have the example of pairs of sister species that occur in the Caribbean and in the Brazil. So these species range from small fish to bigger fish. And what is interesting is that here is a phylogeny of parrotfish that shows mainly in red here that this split of the lineage occur in different moments during the Pleistocene. Some are very recent, occurred like few thousand millions ago, and they are still happening. The speciation process is still happening. Others occur between 1, 2, 3, 4 million years ago. So it shows the nature of this, the nature of this barrier that is intermittent and allows the colonization and exchange of lineages in different moments in the geological history. So here is about the mid-Atlantic barrier. So we have here many lineages that are closely related between, for example, the Brazilian coast, between the southwestern Atlantic and the eastern Atlantic. So we have, for example, the Stegastes fuscus and Stegastes imbricatus that are sister species. We have the Clepticus, that are Brasiliensis and Africanus that are sister species too. And it's interesting to note in that research that explored the phylogeny of the Clepticus clade is that it indicates that the oceanic islands of Brazil might be working as a stepstone for the colonizations of different provinces. So species that are endemic to Brazil, when they reach oceanic islands, they have some opportunities to step stone to jump and colonize different provinces and become different there and so contributing to the overall diversity of the Atlantic Ocean. So here, for example, we have phylogeography of Chromis multilineata, and it shows like an interesting pattern where we have in the left here lineage that are exclusive to the Caribbean, lineage that are exclusive from Brazil, but some that are exported back to the Caribbean and other exported to the Eastern Atlantic. So when we look to the gene flow or the number of migrants we can see that today, currently, the Brazilian province that is considered a peripheral province compared to the Caribbean is exporting towards the Caribbean and towards the Eastern Atlantic much more individuals than vice versa, for example. So it shows the importance of these peripheral provinces to the exportation, to the exchange of the biodiversity between different provinces. And this can contribute to what is known as a biodiversity feedback process, where species ended up migrating from the centers of biodiversity to peripheral provinces. They become different there, sometimes different species, and then migrate back to the center of diversities. So increasing the general diversity of these centers. So it is known as biodiversity feedback, and it occurs in Brazil, in the southwestern Atlantic, contributing to the diversity of the Caribbean, and also within the southwestern Atlantic, as we'll see later in this presentation. So we also have some examples of paleoendemism. For example, here we have two species, both found in the Vitória Trindade chain that is situated offshore in the central Brazilian coast. And you can see here that in the left, the wrasse is only found in the Vitória Trindade chain, and its sister species is found in the tropical Eastern Pacific, on the other side of the isthmus of the Panama. And here is Pemphigus gasparini, that is a species that only found in Trinidad Island, and its sister species is found in the Caribbean, and we don't have any lineage found along the Brazilian coast. So there are two examples of those extinction process that happens along the continental coast, on the mainland coast, and it shows that the oceanic islands, they could be working as museums for these old lineage, mainly those islands in the southwestern Atlantic. So now, looking into the barriers and the evolutionary process within the southwestern Atlantic, we have a great diversity of processes going on there. I can mention here the oceanic islands, where peripatric process can happen, species that colonizate, populations that colonizate the islands, and started to suffer process of adaptation, bottlenecks, and speciation. We have vicariance going on through the Francisco River plume, and we have also some ecological parapatric speciation process, where these colder subtropical waters here shelter different species than the tropical waters found in the northern Brazil. So it's interesting to note that these barriers also contribute to shape different compositional scenarios, compositional patterns along the Brazilian coast. So we can see here that the oceanic islands, they are very different from each other and from the coast of Brazil, but the most different sites are the southern Uruguay and Argentina here. These sites, they shelter very different assemblages, and are very different from everything else that's found in the Brazilian province. So it's interesting to note that the split in the coast is found here in northeast, where the São Francisco River mouth is situated here there, and here we have a strong transitional zone, biogeographic boundary that's found along the Brazilian coast. And we can find in these different sub-provinces different patterns of richness. These numbers represent the number of species found in each one of these sub-provinces, and the numbers between parentheses are the exclusive species. So we can see here that we found the larger amount of species in the central coast of Brazil, and also the larger amount of exclusive species occur there. When we take a look in the number of endemic species, we can find that this province, the sub-province, the central coast of Brazil, shares the highest number of southwestern Atlantic endemics and also the highest number of local endemics, more than 10% of about 10% of the endemic fish of the Brazil is found in this region here. But what is interesting to note too is the number of endemic species in the oceanic islands. So they share a very high amount of species. About 10% of all endemic species too are produced in these oceanic islands combined together. And it's interesting to note that some endemic species are among the oceanic islands. For example, here, these two different complexes of islands, São Paulo's Rocks in the north and Atoll dos Rocas and Fernando de Noronha Archipelago in the south, they share about 8 endemic species between them. And Fernando de Noronha and Trindade Islands, they share 1 endemic species between them that's not found along the coast. So we have here different patterns of connectivity that are not driven by current oceanographic process. They are driven mainly by this ecological factor that connects randomly these species, like with stochastic process that connect these species by the habitat where they live. So here in the Central Coast, I'd like to talk a little bit about the status of the biodiversity there. It's the place where we found the highest diversity in the Southwestern Atlantic. And this is because, as I told before, it's a transitional zone between warm waters that come from the north and subtropical waters that come from the south by upwellings and by a different environment. While in the north we found more coral reefs, in the south we found more rocky reefs, and the community, the benthic community is completely different, and it helps to shape different fish communities as well. So we have here both the overlap between subtropical and tropical species, and we also have this transition, this ecological process that contributes to drive speciation and the occurrence of different species in the southern region of the central coast of Brazil. And this temperature is very important. Some studies that was performed in the mesophotic reefs of the Southwest Atlantic found in the past that some species that were thought to occur only in the central coast, in the southeast, they are actually occurring at deeper depths in the northeastern coast. So here is another evidence of temperature driving the distribution of the species and driving this evolutionary process. And more recently, we found the same at the same depth at 70 meters, exploring different the same habitats, the same similar reefs in the north of the Abrolhos Bank and in the south. We found very distinct communities, and they were related mainly to the temperature. At the same depth, we had a much higher temperature in the north than in the south. In the south, we found many new species with characteristics of the subtropics, while in the north, we found more tropical fish. So it's another example of this transition zone that occurs in the central coast. It extends to the mesophotic reefs and also shows that temperature is very important as a driver for the distribution and evolution of fish in the southwestern Atlantic. So it's not only for reef fish, like we have some evidence found for non-reef fish too, as the macrodon, that have different lineage, different species are distributed according to the temperature, and one lineage is found in the northeast and the other in the central coast and southeast Brazil. So coming back to the northeast here, and we have some this diversity of process going on there. We have, for example, in the south here, we have the São Francisco River mouth acting as a vicariant barrier for the population of these cronies, of these damsel fish. And we also have in São Paulo's rocks, like the peripatric process happening there, where the population is really isolated there compared to the northeast and the oceanic islands that are situated closer to the coast. And we have another example, a classical example in Brazil, of the Vitória Trindade chain. That's a chain of seamounts that are found in the central coast, and they extend from about 100 kilometers from the coast to 1,200 kilometers, where we found the Trindade Island and the Martin Vaz archipelago. So it's very interesting because today most of these seamounts are drawn. They are found, their summits are found around 70 meters depth. But during the ice age, when the sea level were much lower, most of these seamounts became islands. So it allowed the colonization. They work as stepping stones for the fish to colonize the oceanic islands that are situated at the end of the chain. And once the sea levels, with the sea levels getting high again, these populations many populations were extinct to the seamounts, because the seamounts become an habitat much deeper, that didn't allow the existence or the occurrence of the species, the distribution of the species. Many species become isolated in the islands, and this evolutionary process of vicariance start to happen there. So, for example, when we analyze the evolutionary history of the endemic species of Trindade Island, we can see that most of the species are very recent, divergent very recently, and so many of them use the seamounts as stepping stones to arrive there. And many of these endemics, they show scenarios of incomplete lineage sorting, where these evolutionary processes are still going on. They have morphological divergence, but they are still share different, sometimes they share the origin of different haplotypes. And one very interesting pattern that I would like to highlight in this graph is that the seamounts, they work as stepping stone for many populations of fish. As you can see here, like we have haplotypes that are shared by seamounts by many sites in the continental coast and the islands, but we also have some lineage that are exclusive, that are endemic to the seamounts and islands of the Vitória Trindade chain. And more interesting, we have some lineage that are found on the coast, that they are originated from the lineage found in the seamounts, showing that the biodiversity feedback, the seamounts can contribute to the biodiversity feedback, where oceanic islands in the periphery of the Brazilian province can originate new lineages, and then these lineages come back to the mainland, to the center of diversity, and they get different there again. And so the seamounts are contributing to this biodiversity feedback that exists not only in a global level, but also inside the province level. Moreover, here we have examples in the same lineage happen with both vicariance and peripatric process. An example is the Malacoptenus, where in blue here is the species that occur in Fernando de Noronha. It colonized the islands many million years ago, but we have here Malacoptenus brunoides, a species from Trindade Island that still share still is nested inside the species that occurs along the mainland coast, showing this incomplete lineage sorting. That's the evolutionary process that's still going on. And so we have strong selection pressure going on in the islands because they are environments very different from the mainland. And so we have this fast morphological divergence, while the evolutionary history shows that this evolution is happening very fast. So about the assembly rules in the Brazilian coast, we try to assess the beta diversity of the reef fish in the different states and different regions along the coast to see if the assemblage were structured by the proximity between the sites, by a stepping stone assembly, or if it was more influenced by the Caribbean as a propagular rain assembly. And we found that the Caribbean is very influential, very important to the Brazil in terms of composition, not in terms in richness exactly, but in terms of composition. So sites that are more closer to the Caribbean share more species with the Caribbean, more species among them, and the other sites that are more distant to the Caribbean share, that are those situated in the central coast, in the southern coast, they share more species between them, and they have different species, more endemic Brazilian endemic species that are not shared with the Caribbean. But this proximity, this assemblage rule works for composition, but not necessarily for richness. We don't have in Brazil a latitudinal pattern of diversity, with the center of diversity situated in the central coast, in the southern coast of Brazil. And here we can see that this composition changes a lot along the different sub-provinces. So the oceanic islands, they shelter a subset of these compositions. Many families are not found very well in these islands because they have some ecological characteristics that constrain their distribution in the islands. The northeast coast, we have some families that are more represented in the southeast and are not well represented in the northeast. And you can see that there are many families that we found along the Brazilian coast that are not found in the Uruguay and Argentina. So in this research, like we were discussing a lot about the boundaries of the Brazilian tropical reef fish fauna, because some previous researchers were considering that the temperate fauna of the southwestern Atlantic extended all the way up to the central Brazilian coast. And so in this research, we discuss exactly that. That's not necessarily true, because the tropical reef fish fauna actually is extending all the way to the south Brazil, and the families change a lot. We have many, we have even some families that occur at this southern temperate province that is actually not found in the Brazilian province, as Sebastide and as here Notoneridii. So when we analyze the traits, if there are any traits, like try to look for macroecological patterns here and try to understand the assembly rules of the different distributional patterns along the coast. We try to investigate if there were any ecological traits that were related with different distributional patterns, such as coastal species that are restricted to the coast, Brazilian endemics, species that inhabit the islands, and so on. So we found that depth, the ability to use multiple habitats, and the body size are traits that are related with most of the distributional patterns that we found in the Southwest Atlantic. They are very important attributes. For example, here for coastal restricted species, coastal restricted, they have like for species that are widely distributed, not coastal restricted, they occur in much deeper, a higher proportion in deeper zones. They have, for example, here more like generalist species, and species that are restricted to the coast are more specialists. And species restricted to the coast, they have like smaller body size, while species widely distributed have larger body size, and so on. And the opposite occurs in the Brazilian endemics, where most of Brazilian endemics occur in very shallow waters. They are more habitat specialists and have very small body size. And here are some examples of many of the endemic species that are found in Brazil. So we found similar patterns when we were recently studying the Vitória Trindade chain there. We found similar assembly rules determining both the composition and the density, the abundance, the community structure of the fish along the Vitória Trindade chain, where species that have inhabited multiple habitats have more ability to raft. They are not dependent of brackish water. They have higher mobility, higher body size, higher maximum depths. They have the ability to occur and to be more abundant along the Vitória Trindade chain and to reach more distant sites. So all these changes in environment and this evolutionary process that shapes the composition of the species, they are also influenciate the trophic guilds, the community structure along the Brazilian coast. And here we have, for example, a graph that shows that in the sites in the northeast, we have a higher proportion of herbivores, and we have a higher proportion of planktivores. And while in the southeast, we have a higher proportion of omnivores, for example, fish. And shows also that mobile invertebrate feeders, the trophic group that's found in all Brazilian reefs that predominates today. And also the feeding pressure of these functional groups on the substrate, they change considerably along the Brazilian coast, where herbivores, scrappers, they are more abundant, and they have a higher pressure over the benthos in the northeast. We have a higher contribution of omnivores, mobile invertebrate feeders, and territorial herbivores in the south. So to finish, as a take-home message, I'd like to highlight that the southwestern Atlantic biodiversity evolved in isolation with high rates of origination and extinction compared with other provinces and oceans. There is a great diversity of biogeographic barriers shaping the biodiversity of this within the southwestern Atlantic and among the different provinces of the Atlantic, shaping endemic species in the Atlantic that are in the southwestern Atlantic that are not found in the eastern Atlantic of the Caribbean. And the southwestern Atlantic shelters a secondary center of diversity that is driven by this overlap of tropical and subtropical species, and also those parapatric ecological processes that are driving the origin of endemic species in the subtropical waters of the Brazilian coast. So before finishing, I would like to highlight that I'm in a new position at the Center for Marine Biology at the University of São Paulo. I'm affiliated now to the graduate program in zoology of the University of São Paulo, and we have funding for master and PhD degrees. We have funding for fellowships there. So we are working with a global data set of mesophotic reef fish assemblages. So any like student that is interested in any possibility of position, feel free to reach me out. And thank you again for inviting me to talk a little bit about our research, about the patterns of the Southwestern Atlantic. And I will be here to answer any questions. Thank you.
Thank you, Hudson. Was great talk.
That's amazing.
Come on, let's see what Cesar have to tell us. But will be really lovely to try to discuss at least a bit about the ecological drivers, about the macroecological drivers that are shaping all this diversity. So, good luck, Cesar. Thanks for coming.
Thank you. I'm gonna start sharing my screen.
I put you in the present mode. On the left, you can see a sign of more that you can hands up. Right.
Okay, I think something happened here. Sorry, guys.
Don't worry.
Come back now. I'm gonna share my screen. And okay, it is on.
Yes. Okay. I'm listening. You okay? And see your screen, your time. Good lecture to you.
Okay, great.
Backstage. So.
Okay. Thanks, Jones. Okay, thanks again. Good morning. Good afternoon, everybody. So I'm very happy again to presenting here today. I will take advantage of a few things Mariana and Hudson already said, so I will briefly skip something I have prepared, and we can get some time in the end for questions. So today we're going to talk about a little bit about diversity patterns of sea urchins in marine forests. So we saw a lot of maps today, this morning, so this is another map. And when we look at maps with information, we try to check for patterns. So this one is terrestrial patterns for changes in terrestrial vertebrate diversity. So the first thing we can see here is that there is some concentration of warmer colors around the equator and tropical areas. So these are the places with tropical forests, and they concentrate most of diversity of vertebrates. And those patterns, they are driven by geomorphological characteristics and climate and the whole evolutionary history, as already Mariana and Hudson said about the marine taxa. So when we jump in the water and we try to take a look at the same patterns, if we find any latitudinal or longitudinal patterns, we can see for a few groups that are well studied. Like here we have some diversity maps, some species density and functional diversity for reef fishes. And we see that we basically found similar patterns here for species density. We can find higher values around the tropics. And the same is followed by functional richness, that it's basically telling us about ecological roles measured up from attributes. But if you look at the species evenness, we can also see that in the same area where we have higher richness, we have lower evenness. And that means that we have lots of species with less dominance in the species. And finally, we have here the functional diversity that shows a different pattern. And what is special about that? That functional diversity is highlighting here that in a few places, like in the tropical Eastern Pacific, some parts of Caribbean, we have some species that have different functions above the what is expected. So the functional part of diversity is something very important, and it was already highlighted by Mariana, so I won't get too much in details here. And if we zoom into the groups we're going to talk about today, and if we're talking about the Echinodermata, that are a very ancient group, a marine group, and they mostly occur from the intertidal to the abyss plains, so they are very well distributed in marine habitats. And this map here shows the species richness for Ophiuroida groups, and these groups are mostly associated with non-consolidated substrates. So if it follows the same latitudinal pattern that between the tropical lines here, we found the highest diversities, but we can see that in the North Pacific and South Pacific, there is it's a higher diversity, more than will be expected if we compare with another groups. And also because a few echinoderms, they have a high endemism in deep waters, especially in the tropical Pacific. So we can see here that most of the patterns are recurrent for other marine groups, but we have a few details that change for echinoderms. But considering sea urchins that are one of the 5 classes within the phylum, that's the Echinoidea, the sea urchins, those round spiny guys here, that's which we'll be talking about today. And within the sea urchins in the Echinoidea class, we have around 1,000 species, and we'll be talking about 4 orders out of 5, because the infraregularia, the infraclass irregularia, the sea hearts, and the sand dollars, they are mostly associated with non-consolidated substrates. So here I'll focus on shallow waters, especially where we can find consolidated reefs. So that's the places where sea urchins had the most important role. And the classical example is the functional role of sea urchins in kelp forests, where they can be found in higher numbers. So here we have an illustration of some stable states of kelp fungal forests. So we have dominated by macroalgae, those laminaria, those large macroalgae. And this state here, it holds a lot of biodiversity associated with it, a lot of production. But when the numbers of sea urchins rise, and then they can bring this to this state, and those two are stable because the sea urchins in large densities, they can keep this barren dominated habitats, only habitat by sea urchins, a few sea stars, and encrusting algae. And they are very voracious feeders, so that's why if there's no control on these populations, the return to forest dominated state is not certain. A classical example of this was written by James Estes for the Aleutian Archipelago Islands. So in this work, he describes the dynamic of those kelp forests and the importance of sea urchins and sea otters to this dynamic. And since the 17th century, 18th century on, the sea otters were hunted for trade, and then they got almost extinct from for these areas. And they are the principal, like they are the main predators of sea urchins. And when they're gone, the whole community changed to a barren state where the sea urchin populations bloomed, and then all the microalgae were gone. And there is a simplified habitat, and a lot of biodiversity was lost. But in a century ago, when they start prohibiting the hunting of sea otters, then the populations of predators, the sea otters, grow back, and then they start controlling the sea urchin population, and then the forest could grow again. And a few after later, and then with the overfishing of salmon a little bit north, the killing whales start going back to these areas, and they start eating on the sea otters. And then in a few places, this return to a barren state is happening now. This is only the trophic side of it. We are not talking yet about climate change influence this. For the tropical areas, you also have similar work talking about the functional role of sea urchins. So in the Caribbean, there was a huge die-off during the 80s and 1980s, and where the main species of sea urchins are most abundant, the Diadema antillatum, died mostly from all of the Caribbean. And here we can see in this map, it's basically the map of the currents within the Caribbean, and the die-off followed the currents. The die-off still today, it's not sure how it happened, or what's the cause, but it's probably some disease that affected this species. And here in these graphs, we can see the density of sea urchins that were about 10 individuals per square meter, dropped drastically in the beginning of the 80s to less than 1 sea urchin for 100 square meters, and that have a great impact in the macroalgae cover that rose very steeply during the 80s and 90s, and was followed by a decrease in the coral covers here in this plot here. So in the shallow areas of the Caribbean, the role of Diadema was very important. And of course, there was another factors like the overfishing of other herbivores. But in that situation, the loss of sea urchins was very important and changed the whole picture for the Caribbean reefs. And also, sea urchins are considered a delicacy for many people in the world. And in a few places, they have been collected for consuming or for trade. And this could be a problem for tropical areas where the other herbivore groups have been overfished. But in some places or in temperate areas could be a solution for controlling the rising population of sea urchins. And of course, considering the ecological and economic importance of sea urchins, in spite of that, we still don't have well-studied patterns of distribution of sea urchins. And that's what gave me the main idea to start this work. And for that, I tried to investigate microecological patterns of sea urchins, regular species basically, and using open database from different sources, from OBIS, GBIF, and Data1, and different databases, collecting a lot of occurrences, mostly based on occurrence of species. Also references that didn't have databases available, most like older articles. And also I insert a few unpublished data from myself and from collaborators. And from those data, we have to filter the occurrences and species. So I have to check valid species, valid records, because not all database were updated, or there are some occurrences found associated with the museum's coordinates, not actually where they were collected. So there are some faults in open databases. That's something that most of repositories are working on. And also with species, I have to filter only the extant species, because there are a lot of occurrence of fossil species for sea urchins in general. And also I put a limit for species occurring in the marine shelf, so above 100 meters deep. And also working at the species level, not subspecies or subgenus. So it's working with species level. For that, I use the ecoregions of Spalding, using the same hierarchical steps with ecoregions and provinces and realms. And I use those ecoregions to cluster the occurrences. And finally, I applied a few statistics to check out these patterns. Well, talking about the species richness patterns we found, we can check here in this graph. In the map here, it's somehow similar to another marine species, where we can see here, it's, and in the upper graph here, we can, there are the mean values for, the total abundance of the total number of species per ecoregion here in the longitudinal gradient, and here in the latitudinal gradient is the projection of those occurrences here. And the first patterns you can see here is that there is a higher diversity here in the tropics and the equator. We can see here. And if we check the longitudinal pattern, there is a small hump here. The first mode, it's around this part here that matches with the Caribbean area. And the second one, and raises the number of species towards the center of diversity of most reef species, reef-associated species, that is here in the Coral Triangle in the Western Pacific. And if you check the latitudinal patterns, there's also a higher diversity here close to the equator, which matches the Caribbean and the Coral Triangle, which is here between Philippines, Indonesia, and Malaysia. But we have a second mode here that it's out of the tropics and matches approximately the areas of South Africa and Southern Australia and New Zealand. And this is a different pattern from we see for most of shallow marine species, except for a few bivalves and some algae species as well. As I told you before, these are recurrent patterns for most of shallow water species, shallow marine species. And here in the left, we have a couple graphs showing species richness for several taxa. That includes fish, coral, bivalves, gastropods, and corals and other groups. And we can see the same patterns, higher diversity here, one hotspot in the Caribbean, and then another one in the Western Pacific, and a steady gradient for both sides. And for only coastal taxa, we got pretty much the same pattern. And as I showed before, for the Ophiuroidea, another group of Echinodermata, we have the same pattern here, found a large richness species in South Pacific, especially Southern Australia and New Zealand. Part of this higher diversity associated with the Coral Triangle is explained by the historical changes and the continent shapes. And here we have a paper that's very interesting about that from Leprier, and it shows that around 40 million years ago, the center of diversity for the corporate coral that matches the reef associated species diversity center was here in the Tethys Sea, that it's the same place around the Mediterranean nowadays. And this center was hopping, this hotspots of diversity was slowly changing towards the Red Sea and then reaching the actual configuration here in this part of in the Pacific. So the changes in the complexity of habitats here was changing with the currents, we're changing with the sea level going up and down. And this region here was special because it was very diverse in habitats. And those changes in sea level created a lot of different habitats. And this is one of the explanations for such a high diversity in this area, especially in shallow waters. And that's where the coral communities went changing within the tropics. And this pattern is followed for most of the species, the tropical species. Okay, yeah. When we check similarities between the provinces where we found the sea urchin species, we can get to two large groups. One group here, it's composed by the Atlantic and the polar seas, either North or South Pole. And the other group down here, it's formed basically by the Indo-Pacific provinces, either tropical or temperate, and they're closer here. Right? And within each one of these, we have another three, we have 2 domains, 2 main realms, and 6 provinces inside both domains. So when we check the species richness in each province, the first 3 here, the Atlantic and polar seas, they have a lower richness comparing with the other 3 provinces from the Indo-Pacific. And we have a high variation between the ecoregions here, in considering the richness species in Southern Australia and New Zealand. It's something different from the expected. And when we check the Southern Ocean, it has a composition that's quite different from the rest, with a higher contribution of Sideroida. Sideroida, it's an older clade of sea urchins. I'm going to talk about that a little bit further. And for the other provinces, the composition of suborders it's very similar, except for those still here in the Atlantic and Mediterranean. They are composed by a larger part of Arbacioida. That's also older clade, and that's important here because these clades I'm gonna explain better a little further. But as Hudson told before, they are present here in the Atlantic and Caribbean. They evolved isolated from the rest of the ocean. For the Siderid, that's the most outstanding pattern, we can find here in the first graph. They have higher diversity in the southern oceans. And this could be explained by the historical changes in the configuration of continents. When about the same age of 40 million years ago, the Antarctic continent got isolated from the other continents, and we got the creation of the circumantarctic current. That's a strong current. That's compared to other oceanic currents, it's a very fast current. So it traps the larvae of many species from this area here, and this it's another barrier for the dispersion of species here from the Antarctic Peninsula and the Antarctic continent in general. And an important fact for the Psidaridae species that a few species within this class, within this family, they brood their eggs between the spines, so they don't have actually a pelagic phase, so they don't disperse that much. So this is one of the things that maybe had facilitated the radiation of the group within this area. So they couldn't, those species that couldn't disperse that much stay there, and that also there are less predators in general for sea urchin species in this area. So that's why there's a large irradiation of this family in the Antarctic continent. When we talk about the age of the clades, that's I'm talking all the time here about history of these groups. We have basically among the species that are more abundant and they are more diverse in abundance as well. In the seas, we have these two groups. We have some old clades that have more than 100 million years of history, evolutionary history. In general, the Echinoidea had more than 200 million years of age, the whole group. And we have here the Psidarini, Diadematidae, and the Arbaceidae. There are families that they are very old. In contrast, we have some Camaradonta taxa that are very recent, and that they match the age of the last great change in continent shapes. And they have had a high radiation in this period here. If we take a look at the diversity of these two families here, we got Arbaceidae and we got Diadematidae. They're very common in reefs, especially Arbaceidae here in the Atlantic and the Mediterranean. And you can see when these species were formed, the continent was much closer, and the connectivity between the areas were much larger. And when we look at this pattern here, that has very different and very apart hotspots of diversity for the Arbaceidae. That may be explained by a slow evolution, not much change along the time, but that would resemble a very ancient formation of the continents. But for the Diadematidae, there are recent species another old clade, but less diversified. They are found here in the recurrent pattern for reef-associated species. So they have higher diversity here in the coral triangle, and that decreases towards the Atlantic. For those clades with a recent diversification, we got one example here for Echinometridae that follows the same pattern. So recent diversification for the groups, especially in the tropics. There is one problem with these groups for Echinometridae. They are very genetically diverse. So for the Indo-Pacific, there are a few species here that still need to be better evaluated for check those species. And the Estrogilocentrotidae, they are a relatively new taxa. They are found only in the North Hemisphere. And that gives us one idea that the hotspot of diversity is here in the North Pacific, and they cross the whole Arctic, and they also can be found here in the Atlantic, the North Atlantic. This could their evolution, and in fact, in the phylogeny of the group, we can see that the ancient species, the ancestral species were found here in this area in the North Pacific, and they dispersed all along the North Pole, and then they reached the North Atlantic here. And this can be found here in the, briefly, I'm just telling the story very briefly. So we got more older clades here, older groups within this family, and the recent ones, the most recent ones, are found in the North Atlantic, which matches this route of colonization of these species and diversification in the North Sea. But they have roots in the tropic diverse area here in the Southern Asia, Southeast Asia. Okay, but what about the marine forests? And the next step, checking those patterns, were trying to see if they matched any functional role of sea urchins. And based on this paper here from Philip Dexter and Shaveling, where they mapped all the temperate forests and places where there was reported any change from between stable states, from kelp to barren states and back to kelp state, kelp dominated state, or from kelp to barren, then stay as barren, in places where that was not recorded at all. I extended a little bit this map and added data for a few reports in phase shift for tropical areas as well. And here, when we overlap the species richness of sea urchins and this change between stable states, what we can see is not a clear pattern, but in general, in places where we found persistent barrens, especially here, they are also associated to low diversity areas. And in a further explanation, I checked different aspects of diversity. So I checked functional richness and taxonomical diversity as well. So for the first two, I couldn't find any difference, any significant difference, but it was interesting to find that in places where the reefs return to a kelp-dominated state, we found a higher diversification among the taxa. So I got less related taxa in these places. And a further investigation using the functional space of the 21 species associated with phase shifts, I could find 4 different functional entities for sea urchins for those 21 species. And the main characteristics of the species associated with persistent barrens is that these taxa here, they are herbivores. Most of sea urchins are treated as herbivores, but in fact, some of them are carnivores and some are omnivores. They are very plastic in their diet. But all species associated with persistent barrens were omnivores. The long-spined ones, they are basically diadematid. They are that old clade. They are associated with tropical phase shifts. The small-bodied sea urchins, they are associated to transient barrens. So in areas where we have a change back to the forest state, we found smaller sea urchins. And lastly, we got this fourth group, the shallow water sea urchin species. They are associated with all bearing conditions and kelp conditions, and also coral dominated conditions, because this includes the tropical domain as well. But something interesting here, if we check the phylogeny of the groups, most of the species associated and the hominivore species and the sea urchins, they are recent. They have a recent evolutionary history. So, and these numbers here in parentheses, they are the total number on the right, total number of species within the family, and the number of species associated with barren habitats, the formation of barren habitats. So what we have here is that most of the species that have some attributes associated, most of the species associated with barren habitats are recent, and most of the attributes associated with formation of barrens are present in these groups here. They are also found in ancient groups like Abbasid and Diadematid, but are predominantly found in these recent groups here. And this gives us some tips about where the attributes here could evolve, if they came from an ancestor for all these groups, or if there was something that was developed here, just irradiate to all these groups that are close to each other. Well, for future challenge, working with this database, we have to better distribute efforts to sample species, not only sea urchins, but all species of tropical, and especially in the global south, because that were the parts where I couldn't find many data, and that could have biased the results, especially in tropical areas. And also there is no temporal data for many places, because historically most of the studies were done in the north hemisphere. So the historical chains in dominance are better described for these areas here. Another question especially for sea urchins, is that the bathymetric resolution of the occurrence is not great, and of course deep species were kept out of the analysis. But some sea urchin species, they can occupy from the intertidal to down to the 2,000 meters, and the same species can do this. Like Diadema antillarum, that can be found in very shallow reefs and down to 400 meters deep. And lastly, I could only work with the hard attributes of species, the hard traits, because I mostly use morphological traits for that. And refining the attributes used to separate species will be much better to check if really there is one or a small set of attributes that would determine if a species is more prone to create barren habitats, either than only external factors like overfishing, climate change, and etc. That would help understand the whole dynamic of reef areas. And finally, I'd like to thank again organizers, Marina Cecchini, that's not here with us, sadly, and Professor calorosa for the invitation, and also my colleagues that provide data, and those that I know and those that I don't know, but they freely put their data available. So this is very important for science in general. So data availability and transparency in data, everything like that. And thanks for the financing for FAPEG, CNPq, and all my friends. Thank you.
Thank you, Cesar. Amazing. We have a lot of questions, some questions here. And I need to invite back to the screen Hudson and Mariana, before we have a panel
Yes,
discussion.
that's it. I just would like to show some results of
yes,
Marina
you
Piscina.
make a little.
Just to complement a bit, considering all these pictures. But please come, Mariana and Utsu. Let's start our brainstorm here, and let's start to boil our brains a bit, just to have a different plate this morning. Okay, I'm trying to figure out how to fix my brain with. Guys, I have too many computers running at the same time here just because I have the transmission and it's hard. But let me see if I can share my screen where I am. I have a couple of myself to yes, now I'm... Well, regarding seaweeds, wow, I'm really sad because Marina she's a lovely teacher, researcher, well, and friend. So she will be well, I'm pretty sure. She's a bit sick today. So, well, I will try to cover this content just a little bit. Well, regarding seaweeds, yeah, we have a lot to say, but I will be very, very, very quick. Just remembering that seaweed regarding composition, of course, is since looning from the 80s up to now, they are under review. We are increasing our understand about the composition, about the taxonomic composition and distribution. So, yeah, and now we are realizing that all this about geographic information is also important to understand different aspects of ecology or macroecology. And of course, this understanding is also important to understand subjects as carbon, the blue carbon, the role of seaweeds as blue carbon. So, well, as we see in these graphs, this kind of information is really very available and is important to us to understand that we have among drivers, environmental drivers, besides the biological drivers that Cesar and Hudson and Mariana was telling us about, we have temperature, nutrients, and irradiance that together with different values represent a kind of filters that will determine the composition and abundance of seaweed. As we see in this animation, in warm colors, we have higher temperatures. The southwestern Atlantic is a melting pot of conditions. So as we have in the tropical areas warmer waters, we also have the lower abundance or concentrations of nutrients and higher availability of irradiance. On the other side, in the, as I custom to say, warm temperated region, Hudson don't like it really very much. But yeah, we have some key clues that can reinforce the perspective that in the southern of Brazil, we have evidence that we have species that are just present from southern Brazil to Argentina or Patagonia. So from the perspective of seaweed, I think the conditions we have there with high availability of nutrients, lower availability of irradiance, and of course, especially during the winter, lower temperatures, but also during the summer because we have a apualem in all this region. In the end, we have a kind of subtropical environment during the summer, but during the winter with Catalonia and other temperate species, well, we have a temperate. So if we put all of that in a pot, we will have a warm temperated province there. But anyway, this is a discussion for another moment. Okay, and what I wanna show you is that the diversity of seaweeds is huge. We have around 800 species with high diversity of red algae, and we have where Cesar is, more or less, from the north of Rio de Janeiro to southern of Bahia, the higher diversity we have in the southwestern Atlantic. I think Cesar is more or less in the arrow, in the red arrow where I put here. I organized this presentation during your presentation, so it's a bit messy. And here I would like to show you who's Marina in the right corner. Marina, we miss you. I miss you a lot. And this is the results of the main results of Marina thesis. What I would like to reinforce again is that all these environmental factors work as filters, providing particularities that we have in our biodiversity. Here, Marina was evaluating just the coralline red algae molecular diversity. So we have something around 70 motus, taxonomic unities, considering molecular identity. So with these blocks, colorful blocks, we have different unities. So, as I said, the higher diversity in coralline as in the seaweed as a big group, considering red, brown, and green seaweed, these higher diversity sites are in this Brazilian west coast, between Rio de Janeiro and south of Bahia. And what is important to reinforce is that all this diversity is quite important to inform coastal management. So considering that all regions in our coast present some singularities, some particularities, we need to look to this region with special attention. So it's important to keep, for instance, to keep our marine protected areas in southern Brazil, south of Brazil, and need to keep our marine protected areas in our different provinces or eco regions. So as we can see here, we have interchangings between and among these regions, so they are all important. So when we observe the perspective of this flora, we can see that we have three main regions. Hudson knows really very well this paper, was one of the advisors. Thank you, Hudson, indeed. And was very nice. Huh? This process in coral reef was really very interesting. The paper was much better right now. And we have in green the eastern region that represent a kind of transitional region. We have the southern or southwestern region here in pink, and in blue, the northeastern region. In other words, this reinforces a bit of the spouting perspective of two main provinces that you can call as warm temperate and tropical province, and as myself and many others colleagues during decades are saying that Espírito Santo, south of Bahia, from from south of Bahia to north of Rio de Janeiro are a key zone to conservation, to management, etc. of seaweeds. Well, with that, I end. I'm sorry to run too much because we are with our time in our neck. So let's finish this here. I will, okay, not in this computer. I will finish here. Yep, and let's try to start to answer our questions. Can you start, Jones, please?
Yes. We have 3 questions, 1 question for each one, but you can complement the question, the answer to the others. Mariana, you can see the questions on our chat, so you can read too. Mariana, how do you see the near future much better understanding of macroecological patterns being used to practical conservation efforts in Brazil? What's the role reef science is helping this?
Hey, thank you, Jones, for the question from Sergio. I know I've seen the question before. I would like to thank Hudson and Sergio and Paulinho. Great talks. It was really a master class from you guys. It was awesome. I learned a lot, especially for the sea urchins. It was awesome, really. So yes, I see Sergio is stimulating the debate here on the role of ReefSyn, which is our synthesis group, in which I have the pleasure to work with lots of researchers, specialists on reef diversity. And I think we're going to get there because our main objective here is to use these multiple data sets that are available, as Cesar pointed. It's awesome to have data transparency and data sharing, specifically for this project. And we just started to look at patterns using especially richness, but we intend to use, we're also looking at functional diversity, but we intend to take a look at abundance patterns as well to have more information on species distribution, the distribution of diversity, so that we can put this data together for different groups of organisms and better inform management. But I think the main objective of our project is to, and it's a great challenge, is to link the functions of species to their role in ecosystems, and also to the services that reefs provide to human populations in Brazil. And when we can do that, which is the jump from functions to services, then we'll be able to better inform management. I think we're into the decade of the ocean, and we have to walk towards better conservation, but we're missing the human dimension in conservation. And this is one of our objectives in ReefSyn, is to use species diversity, abundance from different groups that form the diversity of reefs, and put this human dimension into perspective, because we are part of conservation. So I think this is what we're going to walk towards, possibly in the next year. I hope that by the end of this year and in 2022, we'll have better answers for that. And I hope that all of you guys can complement my answer. Cesar and Hudson can bring their perspectives into this answer. But it's a challenge.
A real challenge, but we can. Cesar and Hudson, do you want to complement the question? Sergio Rossi, that come now. If you want to complement, please.
Well, I had a question because very recently there was an experimental design in which they exclude sea urchins from a very huge, it was 100 by 100 meters, 10,000 meters square, so it's not bad, the sea urchins. And it worked in the sense that if they maintain the place without sea urchins in a place which was heavily grazed by these organisms,
indeed, the Cistoceira, in this case, another fleshy algae, and also calcareous algae, started to grow up, and also other organisms like sessile organisms, like, for example, metazoans, small bryozoans, hydrozoans, and so on. And it functioned, but I don't know. They just claim that this could be a conservation measure, but I don't know what you think.
In fact, it is. It's kind of sad to say that cooling it's a good management practice, because it was tested in Norway, in Southern Australia, New Zealand, in many other places where the kelp forests are increasing. And it's, I mean, it is not a cheap assessment, like it's not a cheap management, but it works in the long term, because sea urchins are basically regulated by the adult populations. So if we remove the adults, and then we're gonna miss the larvae. So yes, it is a management that it will work.
Just would like to add to the debate. What about the sites where high abundance of urchins are common, you know? Mm-hmm. You guys have found a lot of places where high abundance of urchins are natural, and how to differentiate from places where it's not natural and deserves the management. Otherwise, like, people will start to collect sea urchins everywhere, and in some areas they actually, the ecosystem need them. So
yeah, actually in the Mediterranean there are harvests for food.
Yes.
Historically, but a few species, not all of them, because some of them are bitter, so they go for some species. So usually when we got large population of sea urchins, it's because something changed in the environment. But also in these forests, either kelps or coral, like animal forests, we have patches of habitat. So sea urchins occur in patches because they need each other to protect each other, and then need complexity to live in, so and not being predated. So in general, we have patches of high densities of sea urchins, and these patches can move or be predated, and then they grow, they expand, and they decrease in time. So if you have some external effect, and then they keep growing, and that's the problem. And then we have now overfishing of predators, we have climate change that are already weakening the kelp forests. But we have the opposite in the tropical areas where we don't have much sea urchins, and we need them to control macroalgae.
Wow, nice discussion.
So, yes, like nice discussion. Let's for another question. It's for Hudson and comes from Sergio Flutter. Hudson, what is your guess, or how mesophotic fish biogeography and macroecology will look like related to shallow patterns, or you expect different patterns to appear?
Yes, Sergio, good question. And yes, like, we are expecting a lot of difference, you know, and because the depth, the luminosity, the resource all works with filters for the reef fish community down there. So we don't have that many herbivores or corallivores, and we have a higher abundance and diversity of planktivores and other trophic gills that are more well adapted to those depths. So we have a big change in community structure from shallow to mesophotic reefs. And what we are finding is that when we are assessing different oceans or different biogeographic provinces that have distinct number of species, we are finding that the difference between the diversity between shallow reefs and deep reefs change a lot. And in biogeographic provinces that are relatively poorer, that have less amount of species, the difference between the shallow reefs and deep reefs are not that huge. So we are finding similar values of richness in different biogeographic provinces. So it's really interesting, and we are now discussing and trying to hypothesize that deeper reefs are more saturated compared with shallow reefs. And so when we have like a convergence of similar community structure going on in different provinces, different provinces with different richness levels. So it's been really interesting to study these ecosystems. We are finding a lot of divergence in the richness and in the community structure. So it'd be very, very interesting to explore like all these aspects that you know today of functional structure, functional traits, you know, like how they change between the shallow and deep communities, and how they also change along the latitudinal and longitudinal gradient of diversity.
Amazing, have too much to learn about this. So to the next question was mine. Recent research showed that harmful algal blooms, climate change, and biodiversity loss affect aquaculture production in Scotland and Asian countries. Do you have any information or signs of these economic impacts in Brazil? I ask this because I work with environmental evaluation of ecosystems, and we have scare of studies about this, and I want to know about you and your expertise that we have information about this in Brazil.
Can I add, just because we do have, Jones, these problems also in Brazil. Well, one of the biggest algal blooms of our history was here in the south of Brazil, in more than 2,000 kilometers long of our coastline from the plume of Plata River up to São Paulo state. Is a huge area with Dinophysis acuminata producing a very important toxin, disturbing aquaculture, fishery, etc. And in the northeast of Brazil, we have the Sargassum bloom. So, wow, this is a very global problem right now. Please, guys, sorry.
I'm fine with the question.
Mariana, Cesar, want to start or can I?
Yeah, I'll just add something that especially for sea urchins, some harmful algal blooms can kill the population of sea urchins for an entire place. And that happened in Arraial do Cabo in 2001. And there are a few other signs from time to time that it's coming back. And there are a few mortality isolated here. So it might be important in the future.
Yeah, I could add that all these threats, like all these problems, have a huge impact in our economy, and mainly for those sectors associated with like the marine resource, right? For example, we have the blooms, algal blooms, you know, these get higher in the trophic network. So in several places, you can't eat like those top predator fish because you have problems with ciguatera. So like it's less resource for the economy, like for the local population. Climate change and biodiversity loss are like very well related with like the change in the composition or change in species. We know that biodiversity is related with biomass. Places with higher biodiversity have higher biomass of species, and we have some species that they have a very low redundancy. So if you remove some very important species for the ecosystem, you lose ecosystem services. So Yeah, for sure. Like all these factors, they affect not only the aquaculture but the fisheries, and many other ecosystem service of our coastal ecosystems.
The problem is that these hubs, these hub of algae blooms, which are well known, and there is a specific group worldwide that is working with, are not only more extended, but also more persistent on time. For example, when I was working in Chile, 2006, 2007. In 2006, they had a huge quantity of goniaulax, one of these microscopic algae. It was persistent for more than 2 months in something like 2,500 kilometers, sorry. More or less northern in, I think it was from Iquique and southern part of Antofagasta. So it was a huge coast, much more than 2,000 kilometers in the northern part of Chile, and was a huge problem for the people that also knew that all these algae were just sinking and making the problem of the wrongly said dead zones, because are not dead, but transform completely with this organic material that was respired the whole ecosystem in this case, and the whole habitat functioning. So it was a huge problem, and the problem that we have is this persistence of this kind of nutrients that are going on, continuously, makes this algae much more happier. They are happier, and with the temperature increasing and more, in some places, also more stratification of the water column, they are much more happy. They are happy to be there.
Guys, I need to plug my battery
Where
here
are
just
you,
because
Paulo?
I'm losing my power. But I would like to add that we should also warn that just the global warming can represent a huge economic problem. We have forecast that we should lose something about 80% of global GDP, global domestic product, 80% of the global. We are talking about trillions of dollars when we combine all the threats. So in fact, besides to be a environmental question, it is also a social and economic question. And let's continue. Let's just plug in my computer that I'm almost without battery. Please continue the discussions just a bit more.
You have want to complement something more?
I was going to add a bit to the discussion on your question, Jones, and I was just going to highlight how complex your question is. I'm trying to look at this at the macro scale, looking at the entire Brazilian coast, for instance, and it's before we understand if algal blooms have effects on aquaculture, I think we have to understand the links between, for instance, biodiversity laws and fisheries, biodiversity laws and aquaculture. This is one link. And then we have to see how does climate change affect biodiversity and if it directly influences biodiversity and production. And you see it has multiple levels and it's very complex. It's not only one way or the other, because things here, they affect each other directly or indirectly. So it's a big challenge, and big challenges require good data and lots of data. And here, especially for our coast, and of course you guys can add to this discussion, we don't have, at least for fisheries, we don't have the data we need to assess this properly. So we have to for instance, we have the Cisbiota Mar project, where you guys here have looked at reef sites all over the coast, the Brazilian coast and oceanic islands, which is monitored by PELG. These guys here dive there and they collect information, but we would need information from fisheries dating back at least some decades for us to understand how climate change is impacting species and biodiversity and causing biodiversity loss. So it's a really good question, but answering it requires good data, and we have to get this data together to look at things properly. So if you guys want to add on that, doing that at the macro scale is a challenge. We might have some information for that at the local scale with some studies.
It seems, Mariana, I think that you just put a very, very good, you highlight a very, it seems that we are beggars. And we are always, hey, please give me more money. Please give me more things. Please give me more structure. Please, please. But now, and look, 30 years, no, 30 no, but 22 years ago, I was talking with, in Bodega Bay in California, I was talking with a researcher. And we agreed in one thing. It will be a moment in which will be the contrary. They will beg us to solve the problem 20 years ago. Because we were already aware that there were so many problems, and we don't, I know, please give me $50,000. I need this machine. If you talking with my brother, and I talked with him about a project, I say, well, this is a huge problem of restoration, very serious, blah, blah, blah. This is 20 million euros, something like 180 million reais, which is a huge quantity of money. Okay. And my brother says, that's nothing for Europe. I mean, that's the money that they spend for I don't know, clean the bathrooms in the Brussels. And I would say, that's truth. That's true. Why we have to be always beggars, no, please give me something. And at the end say, guys, if you want some serious thing about the environment, whatever it is, about the sustainability, keep it serious. That's all.
Simple like that. Guys, I'm so sorry, but we are really late again. But I love that, just because we are enjoying a lot and making science something happy. Well, as everybody can see, we are just a simple and big family that do a lot of hard work during the night, during the day, different weathers, different regions of our planet, from poles to tropics. So it's really nice. Well, I love to say I'm working when I'm diving, or I don't know, observing different things. So let's start to end. I'm sorry to say that. And I would like to invite everybody that was or were behind the cameras to come with us, just to say thank you in person. It's not in person, but almost. Come on. Adriana, that was there translating. Maya. Who else? Carolina. Let me see. Here, Natalia, Ravena. And in your name, I would like to say thank you and give you a big hug. And let's do again and again and again, transforming this big planet in a very, very, very small thing that we can hug and make this a very nice and kind of bloom of ideas. Please, this is a moment to say what you want to say. Open your microphone and let's say goodbye. And of course, I can't forget that we need to thanks to Cap Sprint to bring Sergio to Brazil. We are fixing the financial questions, but I think everything will be fixed. Also with the translation process, I would like to thanks to Adriana and Maya for all the help. I think we're going to fix the financial question as soon as possible. And of course, for you students that make that possible, man, you are the guys. So you are our future. So man, please just keep going and studying hard, working hard. This will transform our planet in a better place to all of us. So I'm talking too much. And I also need to remember that for those enrolled on the formular course or on ecology or oceanography, please, I will be waiting for you. I will put the link in the chat at 2:00 p.m. this afternoon. Then we will continue our brainstorm and let's start to put that on paper. Let's write our white paper and say to everybody, to our government, all the message that was discussed during these last 3 days. So please take the words, take the floor. Let's do the finish.
Thank you, guys.
Thank you for everybody. Thank you for FC, for FSC, for the support, for my friends that help for this event happen here. Thank you to every speaker, every lecture that we have. We learn too much. And I believe that the expectation of on the beginning of this event, we get successful. And I'm so happy because this. Thank you for your time. Thank you for time to prepare your presentation, for everything that could support us for our knowledge. We are students, PhD students, but we are students. We have a big challenge on our hands, and we believe that we're gonna be too much with what we learned today. So for me, only I say thank you for you.
I want to say thank you to all of you. Thank you to the program. And this is an opportunity, a fantastic opportunity to know new researchers, which have, we make amazing, really amazing work. So people like Rui, or people like Sergio, or people like La Mariana, Hudson, Cesar, or other people that have been talking during these days. I knew many other people because Martina, or Paulo, Vassallo, these were from my side, but from the side of Paulo came other people. Remember that you have there the opportunity to just inscribe in some work groups if you are interested. You can be from all over the world. No problem with that in the cost action 20102, which is marine animal forest of the world. And it will be a great experience making workshops, making papers together during 4 years. So don't forget that we have this opportunity. Thank you to all of you.
So we can close this event happy. Where Paulo, I believe that you want to say something to the end this.
Yeah, I'm here behind the cameras again, trying to send the message. I'm going to be there at 14. Guys, what I need to say is something very long, but I will not. I will just say thank you. Thank you very much for all of you, and for all of you that was enrolled all these days in this process, for all the institutions. I think now we have much more strong relationships and strong in all the meanings possible. And we are much more closer than ever. Besides the pandemic, besides everything, I think we are learning that we are interdependent and we are vulnerable. And I think science can represent our, maybe one of the few alternatives we have to have a safe ocean and a safe humanity. So thank you for coming. For those enrolled on the graduation course, I'm going to be waiting for you at 2 p.m. or 14 o'clock of this afternoon in the link that I introduced it in the chat. So, see you in the next spring school next year. Bye-bye, guys.
Bye.
Bye. Thank you, guys.
Ciao.
Ciao.
Obrigado.
Muito legal, pessoal.